So humans have a complicated relationship with bacteria. On the one hand, they are our helpful symbionts that are vital for nutrition, digestion, for prevention of chronic diseases, and against colonization of pathogens. On the other hand, of course, bacteria are themselves infectious pathogens. They're leading cause of global infectious disease burden, and antibiotic-resistant bacteria in particular are becoming increasingly common and increasingly difficult to treat. But what makes these distinctions complex are not necessarily all. Bacteria, symbiotic and pathogen, can describe the exact same species. This is best exemplified by the most burdensome antibiotic-resistant bacteria in Europe today. On the x-axis here we have number of cases, and on y the number of deaths. These are the most burdensome in Europe: E. coli, Staphylococcus aureus, and Klebsiella pneumoniae. And all three of these are examples of bacteria that reside as background members of a healthy microbiome, but which cause disease opportunistically, often after antibiotic use. And it's this ecology that makes the epidemiology particularly difficult to understand and to predict. And so the question we ask is, how can we better predict these unique colonization dynamics of multi-drug-resistant bacteria? And we are lab, which is a vital tool in epidemiology and public health. And briefly, this describes the use of using ordinary differential equations to model dynamical systems. And these describe how populations change over time. So, for example, you have a rate of change of population. Methodology is widely used in epidemiology to understand causation, describe transmission dynamics, and make epidemiological predictions. And the most well-known of these models is the SIR model, which describes individuals becoming infected at rate beta at rate. To model antibiotic resistance, however, it's important that our models reflect the actual biology and the clinical reality of what's going on. And what we know very clearly is that antibiotic use selects for resistance in the real world, and so this must be reflected in our models as well. So the classic model for antibiotic resistance epidemiology is the so-called strain competition model. So very briefly, in this model, you have three different types of hosts or patients: those who are susceptible to colonization, those who are colonized with a drug-sensitive strain bacteria, and those who are colonized with a drug-resistant strain. Now, both of these bacteria can be acquired through transmission and through endogenous outgrowth of the bacteria. But in this simple formulation of the model, only the drug-sensitive strain can be cleared by antibiotics, whereas the drug-resistant strain is not affected by antibiotics. And this is really at the core of the mathematical modeling of antibiotic resistance, because sensitive and resistant strains compete to colonize the same hosts, but because antibiotics preferentially clear the sensitive strains, this leaves more hosts available for the resistant bacteria to spread, and it results in dynamics that look a little like this. So on the x-axis, you have the proportion of patients who are on antibiotics, and on the y-axis, you have the prevalence of the bacteria, or the proportion of people who are colonized. And you can see that as your antibiotic use increases, the prevalence of the drug-resistant strain increases as the prevalence of the drug-sensitive strain goes down. Nearly all mathematical models of antibiotic resistance are variants of this typology. There's been systematic reviews in the field to the point where this is considered really the dogma. This is how you model antibiotic resistance. The problem, though, is that this is only one possible mechanism how antibiotics act for resistance. If you speak to a clinician or a medical microbiologist, they're more likely to understand resistance in this way. Instead of it being competition between strains of the same species, we know that in patients who are exposed to antibiotics, their normal healthy microbiome antibiotic. This will deplete the healthy gut flora and allow a drug-resistant bacteria to emerge within the host, and that's the outgrowth of resistance that can occur after antibiotic therapy, usually microbiome recovery, which will protect the patients against outgrowth of resistant bacteria. So our question is, how can we include this phenomenon that's observed clinically in an epidemiological model? And so first, I'll quickly present some theory. We propose a novel epidemiological model for antibiotics. So our leading hypothesis for our model here is that antibiotic use disrupts microbiome function and increases host susceptibility to colonization. So here we have a quick diagram of healthy microbiome at equilibrium, where we have the blue bacteria are our microbiota and the yellow is an invading pathogen. And then on the right, we have the same host but undergoing antibiotic-induced dysbiosis. And we conceptualize that the use of antibiotics affects the interactions between the microbiome and an invading pathogen in three ways. The first is colonization resistance. So the microbiome is capable of inhibiting transmitted bacterial colonies from establishing colonies. When under biosis, this doesn't happen, and so the invading pathogen establishes colonies more easily. The second mechanism is resource competition. So the microbiome competes for limited resource with pathogens, and in a disturbed microbiome, there is less competition, and so the pathogen can succeed more. And lastly is ecological release, where the removal of the microbiome allows internal growth of the pathogen to occur more quickly. So to quickly show what this looks like in our model, in our population model, we have 4 different types of hosts who vary in the status of their microbiome, whether at equilibrium or undergoing dysbiosis, and who vary in whether or not they are colonized with a bacterial pathogen. We have hosts with a healthy microbiome, hosts with a depleted microbiome, and then hosts who are also colonized potentially with the bacterial pathogen of interest. In this model, we model antibiotic dysbiosis as a result of antibiotic treatment, as well as gradual recovery of the microbiome. We also model classic pathogen transmission dynamics, so pathogen acquisition and pathogen clearance, also through antibiotics or just naturally. The key to this model, however, is vary depending on the status of the microbiome. That is to say, hosts with a healthy microbiome are more protected from colonization, and hosts who have a recent exposure to antibiotics are more susceptible to colonization. So how does this model compare to the classical model? On the left is what we do earlier, sensitive and resistant strains. And on the right, we have our microbiome model. And we can see that in this model, we can recreate the dynamics of antibiotic resistance, where with increasing antibiotic use on the x-axis, you can also have increasing prevalence of an antibiotic-resistant pathogen. That and we do this through each of the mechanisms we include. So this pattern can be reproduced through colonization resistance, through resource competition, and through ecological release. So we use a mathematical trick known as next generation theory to derive expressions for R0, for the basic reproduction number, using our model to again demonstrate how antibiotics select for resistance in our model. So here I'm going to show some contour plots where on the x-axis, once again, we have the proportion of patients on antibiotics, and on the y-axis, we have the resistance proportion. So this is how resistant the pathogen is. So at 0, the pathogen is susceptible to all antibiotics, and at 1, the pathogen is resistant to all antibiotics. And we show that as antibiotic use increases for a drug-susceptible pathogen, R0 goes down as you would expect, reflecting effective treatment. And here in this strain competition model, as you increase antibiotic use for a resistant pathogen, you end up selecting for its spread by removing the drug-sensitive strains, allowing proliferation of the resistant strain. So that's for the classic strain competition model, and we show for our microbiome model that we can again reproduce these same dynamics. So as you increase antibiotic use for a more drug-sensitive pathogen, it can still decrease the reproduction number for that pathogen, but conversely, when the bacteria is highly resistant to antibiotics, the use of antibiotics selects the spread of these bacteria by making hosts more susceptible to colonization. So we know microbiome pathogen interactions can independently explain how antibiotics select for resistance. And now quickly I'll show you how we applied this model in a certain jeu de données to evaluate antimicrobial stewardship or bon usage in the French hospital setting. And so our objective is to model to 4 key drug-resistant bacteria in the French hospital setting using data from the literature. We then ran Monte Carlo simulations to evaluate model behavior for each species while accounting for parameter uncertainty, and then predicted consequences of different stewardship interventions on colonization incidents. So very briefly, with 4 different species, MRSA, SBL E. coli, and carbaphenamase producing Klebsiella, four of the biggest concerns in clinical practice right now in terms of antibiotic-resistant bacteria. We drew parameter distributions from the literature, probabilistically for our multi and hospital antibiotic consumption data from the Agence nationale de sécurité du médicament. And here, for example, this just shows the relative use of each antibiotic in the French hospital setting, the most common being amoxicillin and beta-lactamase inhibitor, and the least common of our 18 included class 4 generational spore. Also detail predicting the probability that each of these antibiotics leads to dysbiosis, in other words, that it disrupts the microbiome. And lastly, we included 3 different possible overall amount of prescribing in the population, just reducing antibiotic use. The second is to favor narrow spectrum over broad spectrum antibiotics. And the third is to favor antibiotics with a weak effect on the microbiome as opposed to a strong effect. And lastly, we compared the results that I'm about to present to findings from a meta-analysis of stewardship program efficacy in published clinical trials. So I'm showing one example of the meta-analysis we conducted for MRSA, but we did this for each of the included species. And what do we find? So here in this plot, we have the incidence rate ratio on the X. So this describes after intervention, what is the relative rate of colonization in this population? And so if it's less than 1, it means that the stewardship intervention is effective and there's less colonization after intervention's been put in place. And at the top, we have for all bacteria, and then we stratify by the species. And here we can see that in black here, our models without microbiota predict non-efficacy of antibiotic stewardship. When using real French antibiotic data and the best available parameters, using a traditional resistant model shows that stewardship shouldn't really be effective. In contrast, when we account for microbiome, would we include interactions between the microbiome and the bacteria, and we account for the fact that antibiotics cause dysbiosis in patients, we find much greater efficacy of stewardship interventions, 10% in hospital colonization. And this largely, or at least better, agrees with actual empirical estimates. So these numbers here reflect the overall case across all species, where in clinical trials, stewardship interventions have indeed been shown to be effective for reducing colonization. And is overall, and is a bit messier when you look species by species because of fewer number of studies. So briefly, in conclusion, we present a novel epidemiological model for bacterial colonization, which independently predict antibiotic selection for the spread of bacteria. And existing models to include strain competition and to be adapted to the specific ecology of different species. And then we ask, well, is this model very useful in a real-world setting? And we show that by accounting for multiple simultaneous impacts of antibiotics, we can replicate standard nosocomial dynamics and predict the interventions as compared to a traditional modeling framework. So thank you very much for your attention.